Hello, everybody. Welcome back. Thank you so much for bearing with me through this sort of difficult time. This last couple weeks have been really a big challenge for me. But thank you for being flexible. And hopefully, by the end of Thanksgiving next week, everything will be back to normal. Um, so, for this module, we're really starting to get into the section of genetics, where we're starting to understand how cells and how the machinery within cells then manifests into a physical trait. And that's really what we're going to be talking about today, is understanding the structure of DNA and how that influences um, physical traits, which become things, for example, that natural selection might act upon. So to start off the lecture, I wanted to highlight the work of Dr. Sabanatsu, who has been doing some really interesting studies on the biochemical structure of the ribosome. She's also the lead investigator in one of the talks that I'm having you watch this week regarding the biology of gender. But her lab has really sorry, I lost my mouse. Her lab has really been instrumental in understanding the chemical structure that happens within a ribosome. And ribosomes are where proteins are manufactured, which is going to be, which is really the endpoint of what DNA encodes for. They are encoding for specific proteins. And I wanted to highlight the research that specifically looks at a structure that deals with DNA replication. And you can see here that for each of these structures, you can see what types essentially are encoded for different types of biological processes. And what's critical is that this is really going to be the center point of where protein synthesis happens. And that's where we're sort of going to be leading towards in the next couple lectures. So I thought it would be good to highlight some of her work. So the driving questions for this section are to really understand the structure of DNA and how DNA is organized in cells. Then we're going to be sort of understanding how DNA is copied in living cells, and then how we can actually amplify DNA for use in forensics and other biological fields. So your book chapter this week explores DNA through the lens of the Innocence Project, and understanding how DNA has played an integral role in the study of forensics. But we're going to be focusing on, in the lab, what tools do we have available to actually understand DNA and DNA analysis. And ultimately, that leads us to DNA profiling, and how do we make use of genetic variation in a population to actually create a DNA profile. And then lastly, this is just sort of understanding for an ARC, is understanding how DNA evidence sort of fits into forensic investigations. And this is sort of less important, but it's going to be sort of showing the narrative arc for this next couple lectures. So first off, we have to understand what is DNA. So DNA, from the name, and again, you've probably had this before in previous biological lectures, but what I want you to start thinking about as we're delving into this is, how does this relate to particularly dietary energy, and how cells break down nutrients, which then become integral for other processes in the body. So keep that in mind as we move forward and we start talking about the structure of DNA. So DNA is essentially deoxyribonucleic acid, and that's where the name comes from. So Deoxy is the first part, ribo, and then nucleic acid. And this is really just the hereditary molecule that is passed from parents to offspring. So this is what enables 4 physical and genetic traits that is passed on from generation to generation. And importantly, DNA is common to all living organisms. So when we think, again, thinking back to our previous lectures, everything in that tree of life and understanding the organization of biology, all of them have this common, basically book-like, manual-like coding system of DNA. And it really serves as an instruction manual for how to build an individual, how to create proteins, how to create structures that then create life. So let's think a little bit more about the structure of DNA. So you've probably seen and had before made models of DNA, which is this classic double helix shape. However, what does that actually look like in cells? And how does that become a chromosome? And how are they related? So I thought this was a really useful visualization to understand how this chromosome, which you've probably seen before as this in this typical X shape, how does this shape relate to this double-stranded helix DNA? So this double-stranded helix shape is actually wrapped around these very important proteins called histones. And these histones help determine whether or not a certain section of the DNA is read and created and generates a protein or not. So sometimes you can have, for example, there are studies with genetic twins where your genes are exactly the same, but some parts of their DNA get read while others don't, resulting in different traits in the body. So there are interesting studies, for example, looking at cancer and twins, where one twin will develop cancer where another won't, and asking the questions, why does that happen? And potentially, one of the very exciting regions in biology right now is understanding these histones into what gets read of this genetic information. And how then it actually gets passed down, whether or not this section of the DNA is read or not. And this is an important field called epigenetics, which is something I'm really excited about as well. So these histones help pack the DNA into this sort of closed chromatin formation that then creates that classic X-like shape that you're used to seeing in your textbooks. And we're going to sort of pick up on understanding the 23 pairs of chromosomes that we as humans use, and understanding then how that DNA gets read in the body. All right, we'll pick up in our next lecture soon. Okay.